Hello everyone, welcome to Mastermind. In this video, let us discuss about biotechnology. So this is the second part. In the first part, we have discussed a few basics about biotechnology. This will be important part of our general studies paper 3. So in biotechnology, like I said, the source would be NCERT for us in order to understand the basics. So in the first part, we discussed the definition of biotechnology, and overall we can say that biotechnology deals with the techniques of using live organisms or enzymes to produce products. Then we saw about in vitro fertilization. After that, we saw about the definition which is given by European Federation of Biotechnology, and later on we saw the principles of biotechnology. Under that, we were looking at two important techniques. There were two important techniques for or two core techniques for biotechnology. In that, the first one was genetic engineering. First one was genetic engineering. Second one was bioprocess engineering. Second was bioprocess engineering. In the genetic engineering, it is a technique to alter the genetic material, which is DNA and RNA. In case of bioprocess engineering, it is about maintenance of sterile ambience in chemical engineering. Now, in this video, let us discuss about the proper principles of genetic engineering. Principles of genetic engineering. This will be very important for us in case of exams, especially in case of mains. So let us start from the very basics. Whenever we look at any organisms, they have different strategies in order to produce offsprings. But most importantly, all the strategies fall into two major categories. To get offsprings, we get two major categories. First one would be asexual reproduction, and the second one is sexual reproduction. Which are those? Asexual reproduction and sexual reproduction. Now, what is asexual reproduction? We'll get to genetic engineering once we understand this. Once we understand the difference and what exactly is asexual reproduction and what is sexual reproduction. In case of asexual reproduction, whatever the offspring is given birth, okay, so the whatever the offspring which is generated, they are identical to parents. What is identical? Identical also means same as parents, identical to a single parent. In case of sexual reproduction, here two parents contribute, correct? Here in this case, two parents contribute both genetic information. Two parents contribute genetic information in order to produce unique offsprings. Okay, in case of asexual reproduction, we have identical offspring. In case of sexual reproduction, there is unique offsprings. There are various examples for these. In case of asexual reproduction, we can look at example of salmonella. Now, what is salmonella? Salmonella, I'll write over here on the top right, okay? What is salmonella? Now, salmonella, you have to understand that it is a bacteria, and this bacteria is the reason for food poisoning. Okay, this bacteria is the reason for food poisoning. So what happens is, in the small intestine, a single salmonella cell, there will be a single salmonella cell. So this divides into two. And then rapidly that division starts. Again, it multiplies. So rapidly there will be many copies. there will be many copies of itself. So this microscopic bacteria, this invades our cell and our immune system responds and our immune system responds, and this happens in case of food poisoning. So this causes the food poisoning and we experience fever, nausea, diarrhea, etc. So this is asexual reproduction. So in case of sexual reproduction, we can take an example of humans, and then there is frogs, etc. So this is an example of asexual reproduction and sexual reproduction. Now, why are we discussing about reproduction over here? We are discussing about reproduction because whenever there are multiple genes which are being contributed, in this case two parents contribute the genes, correct? So in this case two parents are contributing the genes. So when this contribution happens, the unique offspring which comes, that will be having both desired genes, that will be having both desired genes and undesired genes. Okay, undesired genes. An example for this. So A and B are parents. Now A may be having the problem related to heart disease. This is just an example, heart disease. So the gene may be carrying some traits of this heart disease. So this also comes to the offspring. So most of the times you can see that doctors will be telling that diabetes has come through genetics, or if one of the parents or grandparents will be having some disease, then that may be carried later on, etc. So in other words, we can say that both types, that is desired genes and undesired genes, they both have come into the offspring. They both have come to the unique offspring. Now, with help of genetic engineering, there is a technique for creation of recombinant DNA. Okay, DNA, because we have seen that genetic engineering is about changes in the genetic information, that is DNA, RNA. So here it is about redoing the DNA, recombinant DNA. In this there is gene cloning, there is gene transfer, etc. Now what happens in this case is, in order to get only the desirable genes, so that the offspring doesn't have any undesired genes, for which we have seen an example also. So this overcomes that limitation. Because here that limitation is there, and traditionally it has been flowing like that only. That is, there will be parents and then there will be offsprings, and the genetic information will definitely come through. But what if there are undesired genes also? That changes can happen with the help of recombinant DNA, in which gene cloning, gene transfer are used. We'll understand what it is. Gene cloning is duplication, gene transfer is transfer of genes, etc. We'll see how that technique works also. Now, in this case, what happens? Only the desired genes has come without introducing undesirable genes in the target organism, which is this offspring. This recombinant DNA, this was first achieved in 1972. Now what it is completely we will understand. So this was first achieved in 1972, and this was achieved by Stanley Cohen and Herbert Boyer. So this was achieved by Stanley Cohen and Herbert Boyer. Now, before we proceed into understanding what is a recombinant DNA, let us understand some basics. Now, for example, there is one bacteria, and this bacteria has multiplied into this, and then later on there has been lot of multiplication, and this has been affecting, let us say, any one part of the body. At that point of a time, our immune system will produce antibodies. When this will also not be fulfilling, at that point of a time, we will be taking antibiotics. Okay, so whenever there is an attack, our immune system itself will produce this antibodies, and then there will be the changes, and then there will be recovery, etc. Now, in this case, we initially had seen an example of salmonella. Salmonella, which is a bacteria which causes food poisoning, that we have seen before only. Now, what had happened at that point of a time? In 1972, Stanley and Herbert, they accomplished this recombinant DNA by isolating antibiotic resistance gene by cutting out a piece of DNA from plastid, which was responsible for conferring antibiotic resistance. Now, this salmonella, a part of this salmonella, okay, which is the circular extra chromosomal DNA, which is plasmid. Now, let us take this plasmid. What is this plasmid? This is automatically multiplying, replicating circular extra chromosomal DNA of this salmonella, salmonella, which is bacteria which with regards to food poisoning. Now, this plasmid, which is native, and it is automatically multiplying. Now, what happens is, when this will be automatically multiplying, our immune system also acts, and there will be antibodies, okay, and there will be antibiotic which comes out of our immune system only. But this antibodies or antibiotic which comes out of our immune system, what was happening is, this plasmid was cancelling this also. In other words, this plasmid was not ending even after the body was producing antibiotic. At that point of a time, what happens? The body, whatever antibiotic it is producing, it is not enough to stop this food poisoning, and then there will be lot of fever, diarrhea, nausea, etc. It leads to death also. Now, this plasmid is cancelling all this antibiotic resistance. Now, at that point of a time in 1972, what they did is, they cut out a piece of this DNA, this plasmid. They cut a piece of it. So let us consider this as an example of a complete plasmid. They cut a part of this. DNA. And this was cut at a particular location. And this discovery itself came to be known as something called as restriction enzymes, or molecular scissors. This is very, very important, because we can read about Nobel Prize also with respect to this molecular scissors this year, previous year, etc. Molecular scissors. So this cut piece of DNA, whatever is cut, now this is part A, this is part B. This cut piece, let us take only part A. This cut piece of DNA, this was linked with plasmid's DNA. This was linked with another DNA of plasmid's DNA. So a DNA is taken and a specific location is cut, and then to that the plasmid's DNA is attached. Now, what happens when this is attached? It acts as a vector. Because it is then released in the body. Previously, it was cancelling this, but later on, because it is from the plasmid itself, it will not be able to cancel that, or it will not be able to resist that. And with that, we'll be able to provide the enzymes also. We'll be able to provide the medicines also. In a real life scenario, you can consider a mosquito. Okay, you can consider a mosquito. This mosquito carries this malaria parasite. This mosquito is carrying this malaria parasite, and this inserts it in human body. and that is how humans get malaria. Okay, so this is a vector which is carrying this parasite and it is inserting in the human body. Similarly, in this case, this whole thing becomes a vector. This whole thing becomes a vector and then transfers it into the alien piece of DNA which is in the host organism, and with that the in vitro DNA, which is this DNA, which is mixed, right? So this linking of antibiotic resistance gene with the plasmid vector, this becomes possible with the enzyme DNA ligase. This combination itself is possible because of DNA ligase. So this is possible with the help of DNA ligase, and this acts in order to cut DNA molecules and join their ends. So this makes the new combination, which is this new combination. It has half of the one thing and half of the plastids thing. So this new combination of circular autonomously created DNA, which is in vitro, this is known as recombinant. This is known as recombinant DNA. Why was this created? Because previously the plasmid was not allowing the antibiotic which was coming inside the body only, which was inside the body itself, it was resisting, and hence to that plasmid itself another DNA was joined, so that it acts as a vector and it goes in vitro into that host organism and then starts to multiply. With that help, we'll be able to destroy this bacteria, and hence cure the humans. So this is known as recombinant DNA. Now when this DNA is transferred into this Escherichia coli, which is a bacteria closely related to Salmonella, it could replicate using the new host DNA polymerase enzyme and then make multiple copies. So this itself is known as DNA cloning and recombinant DNA. Now there are various steps which we discussed in just genetically modifying an organism. Various steps, and that can be studied in three parts, that can be understood in three parts, which you have studied now. First one is, we have to identify the DNA which is desirable, which is having desirable genes. Here we have seen that there are desirable and undesired genes, right? So we have to identify which are desired genes, desirable genes, and then they have to be introduced into the host. Okay, this DNA has to be introduced into the host. Then this maintenance of introduced DNA in the host and transfer of this DNA has to occur. DNA desirable has to be selected, then in the host DNA has to be inserted, and then that has to be introduced into the host, and then that has to be transferred in the host DNA. Okay, so these are the three steps which happens in genetically modifying an organism, which happens in genetically modifying an organism, and then we have genetically modified organisms. So we have seen genetic engineering, bioprocess engineering. In genetic engineering, we have seen asexual reproduction and sexual reproduction, and we have seen an example of Salmonella. Then we saw there is recombinant DNA, which is cutting off one thing and then joining it with the desired DNA, which is joined with the desired DNA. In this case, it was joined with the plasmid, which acts as vector, and then it goes and multiplies, then it cures the organism. So I hope you have understood about this. In the next part, we'll be discussing about the tools of this recombinant DNA technology. What are the enzymes which are used? What is the sequence? What is the endonuclease, etc. We'll be looking at in the next part. Now, at the end of this e-paper, there are a few multiple choice questions for you to answer. You can attempt those. Thank you. I'll see you soon in the next one.